Yeah. Okay.
Hello, ladies and gentlemen. Welcome to DC News, to my weekly talk show, Outlook. Today I have a very special guest from the Netherlands, Professor Muhamed Amin. Welcome here to DC News. Muhamed
Thank
Sbahlul,
you.
how are you?
I'm fine. Thank you. And yourself?
Fine, thank you. I'm glad you accepted my invitation in my weekly talk show, particularly about such a delicate subject. I came across one of your articles recently, ACE2 coding variants in different populations and their potential impact on SARS-CoV-2 binding affinity. What does this article suggest, and what motivated you to start this research? You're a professor at the Groningen University. And if you could also please introduce yourself.
Yeah. Okay. Thank you for the invitation. I'm happy to be here. I'm an assistant professor at the University of Groningen, Netherlands, and I'm a scientist at the Center for Free Electron Laser in Daisy, Germany. Well, yeah. So this is actually my third article, or actually my second article on COVID. I had 4 articles on this research topic. What motivated me? I had the first article published in May 2020, and I used computer simulation to show how the binding between the SARS-CoV-2 is different from the SARS 2003 virus due to some mutations. So, and also with this study, I showed that it's unlikely that this virus is manufactured by human. So I published this study, and I saw that it has an impact on the field, and many people cited it. So I also thought, okay, so if I study the mutations, I can also apply this idea to understand why different populations respond differently to the virus. We saw how strongly the virus impacted Europe, but in Africa, it wasn't as bad as in Europe. So with other colleagues from other universities in Egypt, I was able to identify the mutations that frequently appeared in different populations. So we take this mutation and put them in a computer program that can do this mutations on the computer, and then we calculate the interactions between the receptors from the cell and the COVID, and see how this affected.
So if interrupting you, now let's go step by step. So there we have the virus, and the virus has a spike. Yeah. And please correct me if I'm wrong with what I'm saying. And then we are looking at a sort of docking mechanism to the cell membrane, to the cell surface. And part of this docking process is influenced by this ACE2. What's the relevance of the ACE2? What does this docking process mean? Before we look at the various genetical forms this ACE2 might be coded, we need to address also the question, why are we looking at it? What's the role ACE2 is playing?
Okay.
So to assure a better understanding for our audience.
Sure. So
and for me as well. And for myself as well.
Okay, thank you for the question. So the idea that you can so the virus needs to bind to the cell in order to inject the RNA so that it could reproduce itself. So if this is the cell surface and the virus needs to get there to bind, it has to stick somewhere. And it uses the protein that exists on the surface of the cell, which is called ACE2, to bind. And this is simply electrostatic. So you can imagine, we studied in high school that if you have a negative charge come close to positive charge, they get together, right? So it's exactly the same idea. It's a bit more complicated, that the charge are distributed through the amino acids, which the amino acids are the building blocks of the protein. Some amino acids carry positive charge, some amino acids carry negative charge, some amino acids are neutral. But although they are neutral, they still carry partial charge distribution. So they still carry electrons moving everywhere, and this makes electrostatic interaction. So the virus protein, which is a spike protein, has amino acid. You can think of this as a potential or electrostatic or charge that are positive and comes next to the ACE2, the ACE2 service, which is what we found out in our first study on the binding between the virus and the cell. The receptor of the cell has a negative charge or negative potential. So they kind of attract each other, and the virus used this electrostatic interaction to get into the cell and inject its RNA inside. So this is the first point. It's a kind of docking we talk about docking if we don't know where the molecule will bind, but we exactly know where the cell comes to bind to the ACE2. This part is called receptor binding domain, and it already has a crystal structure in the protein data bank. So we download this. So crystal structure meaning that the structure is obtained by X-ray. So what we do is we download this structure. We have the XYZ coordinates of the atoms, and then we apply computer programs that calculate the electrostatic interactions between the ACE2 and the virus. And then we are able to say how much it's tightly bound. So, and then it comes to the next step, as you were explaining, which is now if there are mutations in the virus, how, or in the ACE2, how this affect the interaction with the virus. And now we can answer the question if different population, why they would respond differently to the virus.
I understand. Before that, you meant that there's a significant difference between SARS-CoV-2 and SARS-CoV.
Yeah.
What's the main difference? How would you describe this difference?
Well, the main difference are some mutations in this receptor binding domains. So some amino acids change due to the change in the gene of the virus. So these amino acids, where they're changed, when they really change the electrostatic potential, they make what we found in our first study, which is published in the in the American Chemical Society. We found that it's changing a few amino acids really change the whole potential and make it more likely to bind the cell. And this might explain why the 2019, which is COVID, is really has this outbreak compared to the SARS 2003.
And so the electrical binding capacity is different. Is that partly explaining the virulence of SARS-CoV-2? Is that something you are referring to?
Yeah, we found that the electrostatic interaction is significantly higher with these mutations. I think there was like 5 mutations in receptor binding domain. The receptor binding domain is the part of the protein that goes and binds to the cell, the ACE2. We found few mutations there, like 5 mutations, and really affected the binding affinity and made the virus much easier to bind to the ACE2.
So actually this S protein refers to a prion-like domain. So a prion is a protein, a misfolded protein in broad terms, to a prion-like domain, and to the way prions usually enter a cell. So my question is, if the binding affinity is influenced by this protein, could actually the plethora, the abundance of studies related to the cell entry mechanism of prions provide us a better understanding of a so-called respiratory virus? I mean, prion disease are severe neurodegenerative disease. They start with flu-like symptoms, but it's definitely not flu.
Yeah. Well, of course, because if we understand how the proteins bind to the cell, we can design drugs to block the binding process. And this is actually one of the main idea of designing drugs to fight viruses in general. For example, the HIV is the current medication for the HIV, which is called cocktail, is some kind of drugs that you have to take every day so that you block the virus from getting into the T cells and inject its RNA inside. So understand how it works, we can block it. And this is a very important problem in drug design. And now it's called a big field called computational or computer-aided drug design. So if we know the structure, we understand how it gets into the cell, we can find the small molecules that goes and bind to the ACE2 or to a virus so that it block it from binding to the ACE2.
Well, interestingly, the article I mentioned at the beginning of the interview, now again, ACE2 coding variants in different populations and their potential impact on SARS-CoV-2 binding affinity, is somehow unusual. Normally, someone would say that all of us, particularly in a sort of egalitarian world where everybody, all of us are equal, so obviously a virus seems to distinguish and have preferences. Is that something which you say, basically, that we are not all of us might be equal in front of this virus?
We are not equal. When we saw this, we saw that females are less affected by the virus than male. And
that's the first inequality, huh?
True. Yeah, and the idea that we have mutations and we have, so it's always, we are always different how to respond to all diseases, not only one. So that's why you see different people respond differently to flu, for example. So as I mentioned, there are two parts of these interactions between the virus and the protein. These are the S protein of the virus and the ACE2. A change of any part of those would affect how they interact together. So in the first study, we studied the change in the virus itself. But now in the second study, we thought, okay, so how about changing in the mutations of the humans? And we identified mutations in populations, and we told the computer to perform a simulation. What will happen in reality if we have this mutation? How the binding affinity between the virus and the ACE2 will change based on these mutations. And we found
on the one side we have actually the mutating virus, and on the other side we have mutations of certain population groups, and the way they interact with each other. Is that basically what you're saying? At first you look at the various mutations of the virus, and now you say one key to enter the cell is the ACE2 protein. Is that the protein? Yeah, is that correct?
Yeah, it's a protein on the cell in the cell
and
surface.
This has various coding variants. And now you did an analysis in different populations and computational chemistry calculations to probe actually the effects on SARS-CoV-2 with the ACE2 interaction according to the various coding variants. Is that correct?
Is it true? This is true, yeah. What we did is we identified these mutations and went to the structure that we downloaded from the protein data bank and used the computer to replace this amino acid. So the computer kind of searched all possible conformations of the protein, because you are now changing something in the protein that could affect the neighbor amino acids. So the protein tried to find the minimum energy conformation and does the mutation for you, so you don't have to do it in the lab and figure out the structure again. So
that's fabulous.
Yeah, so the computer is really facilitate this study, and it's much easier now with this computational power and supercomputers we have, and also the development in software to do such things. So we use a program that it's designed in State University of New York, and we performed the calculations, and we saw that there is significant differences between the populations, how they respond to the virus.
How many coding variants? There are how many coding variants of the ACE2?
There are many, but the idea that we only selected the coding variants in the receptor binding domains. So, for example, if we have amino acid that it's really don't care about the virus, and it's way far from the binding domain, then we don't really study it. We just study in the part where the virus comes to bind. The virus comes to bind, so it's very.
I mean the ACE2 coding variants.
Yeah. So.
And at how many different coding variants were you looking at?
For each population, we looked at a single mutant. Okay, so for each population, we looked at the amino acid that most likely to change based on the population. We change it and we study the interaction between this amino acid and virus.
And which is the
And we saw that there is a single amino acid could really make a big difference.
And which population, for instance, let's say, has a plus, and which population groups tend to have not such a favorable mutation in terms of the docking mechanism?
Well, we saw that Europeans and European South Asians are really mostly affected with the virus. So let's say to be accurate, they have, based on their mutation, they have the highest binding affinity with the virus. So they likely, it's harder for them to fight the virus, okay? And then Africans respond less to the virus, South Asian less, and also Ashkenaz Jewish also less. So we saw that it's really significant differences between populations. However, I have to also mention that there are other factors not included in this study, and you also pointed to another protein that could play a role.
So regarding the ACE2 mechanism, the electrostatic mechanism, there you explained well which populations might have an advantage and which others might be less advantaged in terms of the binding affinity. Now, in addition to these findings, let's have a look at actually what you mentioned, the second decisive factor for the docking mechanism, which is so-called the TMPRSS2. What patterns, and now first of all, what is TMPRSS2 and what patterns in the different populations do we find here?
Yeah, okay. Well, I didn't really do a study on this protein, but in general, this protein activates the S protein of the virus before it binds to the ACE2. So you need to pass by this protein first in order to do the binding to the ACE2. Yeah, so both of them are important and both of them play a role, but it's really hard to deconvolute. I mean, it's really hard to decide on the total response of the human based on these small contributions from all the proteins. So TMPRSS, I have seen other studies, other papers say that the TMPRSS, with some mutations, it's really, people are really able to fight the virus much better than others, and they also kind of found a different conclusion that the Ashkenazi Jewish they are able to, they are less resistive to the virus. So this is now if it's ACE2 or TMPRSS, which one is the more dominant? We actually don't know. So it's very hard to include all the factors in one study.
But here, some populations have a higher susceptibility than, particularly the population which you indicated in your paper.
Yeah.
Then regarding the ACE2 binding affinity. So could you have, let's say, a population which is in terms of ACE2 privileged and in terms of the second decisive docking mechanism have a higher susceptibility? Is that a contradiction, or is it just by nature that you have advantages and disadvantages in all areas?
Well, yeah, it's the idea that you don't know that if you can have mutations that enhance the binding between the virus and ACE2, but make the interaction with the tempress less, and then the total effect is not easy to figure out. So the idea that it's really hard to figure out the total contribution, but we can publish studies on each protein, how the mutation affect the interaction, and this could give some explanation why we think that this population is respond differently than other. But the binding to the cell, I would say, it's really a major factor, because this is the drug target for all the drug designs to inhibit the binding to the cell.
So we cannot say which plays the larger role in this docking mechanism, ACE2 or tempress. Is that right?
Yeah, well, yeah, but I would say my opinion that ACE2 is more important, and we saw that with the time of COVID, we were not advised to use ibuprofen for fever. We are advised to use the paracetamol instead. And one reason for this, that the ibuprofen was increasing the cell receptor, the ACE2, making the virus easier to bind to the cell. So I would think it's a really dominant effect from the ACE2.
So that's interesting. So your study even sorts out some of the very hot debates during the first days of Corona, which would be the better therapy. And obviously you submit us also the very precious information, how this is linked with your research and this electrostatic binding affinities. That's right.
Yeah. Well, protein electrostatic now is the major and the dominant interactions between all biological materials and the proteins and the drugs.
But if there are such differences between the various populations which influence the binding affinity, will we then also have to deal with various therapies according to the various populations, or does it mean that we need for each population group, according to its genetic susceptibility, different drugs, or we could identify drugs treating all of us equally?
Well, the gene therapy now is something that people study, but in general, you don't have to do this, because you can design a molecule or a drug that binds to the virus stronger than any other mutations. Because you can engineer this, you can do it yourself, you can understand what are the dominant mutations, what are the most important interactions. You can design a molecule so that you make sure that it will bind stronger than any other mutant. So in this case, you don't really to worry about designing a molecule or a drug that works for a population but will not work for the other. But you see, even with the side effects, the side effects change from a person to another. And this also because of these mutations.
At the beginning of our interview, you mentioned that you've firstly started looking at various mutations of SARS-CoV-2, from SARS-CoV to SARS-CoV-2. And you also mentioned that you do not believe that it's man-made. Does this mean that we are dealing with mutations which just simply occur in nature?
Yeah, this is what I think, because when I looked at the mutations and how they are they are distributed in the binding domain, it was really hard to believe that this could be done by human, because they are very complicated. And the idea that the virus already exists in 2003, and there are several mutations took place in this period between the 2003 and 2019. But it happened that in 2019, the mutations really impacted how the virus binds to the cell. So, yeah, so I have no reason to believe that it's man-made.
many people, so particularly those who claim that it was man-made, they associate the man-made virus also with a biological weapon. So I think it would be definitely better having a mutation which simply occurs in nature than having something man-made, because that would mean automatically that it could be a weaponized virus.
Yeah, exactly. And it also affected everyone. It affected all the countries, and there was no selectivity. So why would you do something like this? And I saw people, really old people, survive the virus, and many young people did not survive the virus. So what is the idea of designing such a virus? So there is no sense of thinking like this. But this is my opinion.
Well, but at least the more arguments we find that these mutations appear in nature, they simply occur like that, the more comfortable is the information, because we then don't need to be scared of a biological weapon. I don't know if you share my view.
Yeah, I don't think you need to worry about this at this moment.
Yeah, but still, a perfect biological weapon would be one which would distinguish between the host and the enemy, because a biological weapon should not kill everyone, should just kill according to a pattern, or should kill the enemy, whoever the enemy might be.
Yeah, but the idea is that even with these mutations in population, they are not in 100% of the population. If they are in 10% of the population, then this is a lot. So it's really hard to design such thing, because even there is nothing defined the population in the DNA sequence that makes you comfortable with designing something like this.
I will have an interview very soon with Professor Eric Xavier Almeida from the University of Sao Paulo. Yeah. He's an expert in prion disease and also a famous author about the weaponizable aspects of prions.
Okay.
So I'll definitely look in future at this aspect as well. I read a few of his papers, but it's far too much for me to get a perfect understanding of what it's all about. Yeah. But I think that a biological weapon would work professionally if the pathogen agent is making a difference. Otherwise, it would kill everybody.
Exactly. So, yeah, as I mentioned, and these mutations are really not frequent in the population. 10% is frequent enough. So I'm sure there is research in this perspective, but it's not my field of expertise. But this is my just an idea or the point of view from just a normal reader.
Could we talk about genetic control, or would it be rather, well, I know it's a matter of semantics, yeah, or genetic susceptibility? Which way should we call it? A genetic control would be something when dealing, let's say, with a biological weapon, because it controls. The susceptibility refers to the host, which host is more vulnerable and which host is more safer against a pathogen agent.
Well, to say it's control, as you mentioned, it's tricky. So I don't know how much of control you can have on this, and it's designing such weapons or such molecules that will select the host based on genes or DNA sequence is really challenging, because you don't know also the side effects, and you might study that that your target is a certain protein, and you design your molecule based on this, but then on the other side you find that there is another protein responding to the molecule, even if you are planning to have it in a certain host, but in another host the response comes from another source, then it's really hard to control it. So, but I as I mentioned, this is just, I'm not expert in this field, so you cannot really, you have to ask someone else.
No, that's right. No, since we're approaching the end of our interview, that was just a final question, definitely not related to your area of research. Well, Professor Amin, Muhamed, thank you very, very much for joining here on a Monday, live to Outlook in my weekly interview here on DC News. That was a pleasure talking to you. Thank you for explaining us what you are studying and also telling us more about this so tricky virus, but also offering us some good reasons why we should not be afraid of a biological weapon. Thanks a lot, and my best regards to Groningen, to the Netherlands.
Thank you very much. I appreciate your time.
Have a good week.
You too. Bye.
Bye.